Biochemistry 401, Lecture 37. Today we're going to talk about transcription, both in bacterial and eukaryotic systems.
Hi, today we're going to talk about transcription. This is the synthesis of RNA through the polymerization of ribonucleoside triphosphates by using DNA as a template. Before we get started, it's important to get some terminology straight so that we all start on the same page. RNA is transcribed from genes. When the genetic sequence is written with two strands, the top strand will be the coding strand and the bottom strand is the template strand. The top strand is called the coding strand because it's the same sequence as the primary RNA transcript is going to be, except that the RNA is going to include U's instead of T's. The bottom strand is the strand that's going to be used for the template for the synthesis of RNA. We also have to have some terminology so that we can talk about directions in relation to a landmark. In transcription, these directions are upstream, which is in the 5' direction, and downstream, which is in the 3' direction. And this is in relation to a landmark. And it's also in relation to the top strand. Why? Because in general, the top strand, the coding strand, is the more important sequence. For instance, when we write a genetic sequence and only use one strand, we write the coding strand 5' to 3'. We don't write the template strand, generally speaking. And so, when we're talking about terminology that includes upstream and downstream, we're talking about the 5' direction and the 3' direction, respectively, in relation to the coding strand, the top strand. I hope that's clear. There are three main stages of RNA synthesis: initiation, elongation, and termination. Initiation is beginning, and in order to begin transcription, the polymerase, the enzyme that's going to synthesize this RNA, has to find out where to start. So it has to find a region in the DNA that's called the promoter. It's going to bind to the promoter in order to initiate transcription. So, promoter recognition, and then we have to form an open promoter. That is melting the DNA strands so that initiation can proceed. In order to begin transcription, the polymerase actually has to leave the promoter, so that's called promoter escape. And then we have elongation, and finally we have termination, when RNA synthesis stops. And so that's initiation, elongation, and termination. We're going to look at the synthesis of RNA in prokaryotes like the bacteria E. coli, and also in eukaryotes, and we're going to use humans as a model system for that. There are similarities and there are differences in these two types of transcription, those in prokaryotes like bacteria and those in eukaryotes like humans. The similarities for both systems are shown here. Both require template DNA that has promoter and termination signals. They both require ribonucleoside triphosphates, and these are A, G, C, and U. We have U's instead of T's, remember. And polymerases in both systems require magnesium as a cofactor. And finally, both systems require a polymerase. But what isn't needed is a primer. Primers are not needed. Since we're using RNA for transcription anyway, we don't need an RNA primer. The actual process of polymerizing this RNA is also similar in prokaryotes and eukaryotes in this respect. The template strand is going to be read 3' to 5', and the RNA itself is going to be synthesized 5' to 3' in a fashion that's complementary to the template. The synthesis of this RNA is going to be driven forward by loss of pyrophosphate and subsequent hydrolysis of the pyrophosphate to yield 2 molecules of inorganic phosphate. It's the same in prokaryotes and in eukaryotes. And another thing is that we need magnesium in order to position these substrates at the right place inside the polymerase and also to help catalyze the reaction itself. So magnesium is required as a cofactor. But there are many differences too. The polymerases that are used in prokaryotes and in eukaryotes are different. For instance, there's one main polymerase for transcription in bacteria, but there are 3 main polymerases that are used in eukaryotes: Pol-1, Pol-2, and Pol-3, and we'll talk about those later. So the polymerases are different. The way that RNA synthesis is initiated is also different. It's much more complex in eukaryotes than it is in bacteria, and termination is also different. Eukaryotes also use a lot of post-transcriptional modifications in their RNA, especially messenger RNA. That's the RNA that we use to make proteins. That's substantially modified. We'll look at that in a minute too. And another thing that's different is that transcription happens in the same place as translation in prokaryotes like bacteria and archaea. That's because the genomic DNA is situated in the cytosol. There's no intervening nuclear envelope. And so the transcription and translation can happen at the same time on the same transcript. As a transcript's being made, it can also be translated. That's different in eukaryotes. In eukaryotes, our genomic DNA is in the nucleus. And so we can synthesize messenger RNA, for instance, but then that transcript has to be exported out of the nucleus and into the cytosol for translation.
So let's talk about transcription in E. coli. This is a prokaryotic model system. And let's look at the polymerase first. This is the enzyme that's going to actually synthesize the RNA. This enzyme contains two major parts. It contains a targeting protein that is called the sigma factor. This is going to target the holoenzyme to the promoter. And it also contains the core polymerase. This is the portion of the enzyme that's actually going to catalyze the polymerization itself. Since the sigma factor is the targeter, this is going to provide the specificity of binding for the polymerase to actually find the right promoter to transcribe the right gene at the right time. And so E. coli has one main RNA polymerase, but it contains several sigma factors, and it's the sigma factor that provides the specificity for transcription for bacteria. The core polymerase is made up of 5 main regions: alpha 1, alpha 2, beta, beta prime, and omega. We're not quite sure what omega does, but alpha 1 and alpha 2 are structural. Beta and beta prime are catalytic. Those subunits are going to be the ones that are actually going to catalyze the polymerization itself. These two round objects that we see on the alpha subunits are the CTDs, the C-terminal domains. These are very important for stabilizing the polymerase on the target DNA. We're going to look at that in just a minute. So how does sigma know where the beginning of a gene is? How does it know where to go? Well, it goes to a region of the gene called the promoter region. And in bacteria, the promoter region has 2 main parts. It has a -10 and a -35 region. What does that mean? Let's talk about that for a minute. We have the start site of transcription. Let's call that 1. That is 1. And then when we're going upstream, we have -1, -2, -3, -4, all the way up, -10 and -35. But please note that there's no 0. There's 1 and there's -1. Downstream of +1 is going to be +2, +3, +4. Upstream is going to be -1, -2, -3. The regions of the promoter that we're interested in for sigma binding are called the -10 region and the -35 region. And the intervening space is also important between them. Different promoters have different lengths of DNA between these two regions. As you can see from this diagram, the -10 region is also called the Pribnow box, and this is what we call the -10 region in E. coli. Both the -35 and the -10 regions are very important for attracting the sigma factor. That's because the sigma factor binds specifically to these places on the promoter. The intervening space is also important between these two regions because it will dictate whether that's a comfortable fit for the sigma factor or whether it's a strained fit. If you notice at the bottom of this slide, there's something that's called the consensus sequence, and this was determined by looking at the promoters of several bacterial genes and saying what nucleotides are the same and what are different. And those nucleotides that were most often seen were noted. And the optimal space between these regions and how far from the initiation site these two regions are were also noted. And so this yielded what's called a consensus sequence, the most frequently seen nucleotides, the most frequently seen spacing. And promoters that are closer to this consensus sequence are stronger promoters than those that have deviations from this consensus sequence. Deviations from consensus yield a weaker promoter. because the sigma factor can't bind as easily to that promoter. And in this side, we actually see the sigma factor bound to the promoter, and we can actually look inside the polymerase to see where the DNA has been unwound a little bit, and we see the template strand and we see the non-coding strand separated. This is an open conformation, and the polymerase itself, because of its architecture, actually does that itself. It actually unwinds the DNA itself to open those strands. Pretty cool enzyme. You can see at the bottom there is a heteroduplex between the RNA and the DNA template, and then we see the exiting of the RNA from the polymerase. And so as the polymerase goes along, it scoops up ribonucleoside triphosphates and it synthesizes the RNA chain, and that exits out of the polymerase. And before and after, the DNA is unwound and rewound. In this figure, we see those two round regions in the alpha subunits, the two C-terminal domains, and those are involved in binding to a specific portion of the DNA called the up sequence. This up sequence is found in some genes and not in others. It's found in some genes that are transcribed in high quantity, like tRNAs, for instance. So this allows the polymerase to be attracted more strongly to the DNA because there's something else to bind to, right? The C-terminal domain in the alpha subunits can bind to this up sequence and make a more efficient binding between the polymerase holoenzyme and the target DNA. And so these types of genes are transcribed more efficiently and more often because they're better at attracting that polymerase. So what happens during transcription in E. coli? Well, the first thing is that sigma factor has to bind to a core polymerase to form the holoenzyme. And then that holoenzyme can find a gene and find a good fit for that gene by locating the promoter. But this is a closed promoter initially, and so it's important to go from a closed promoter to an open promoter. So first of all, we have the pre-initiation complex, which is sigma factor and the polymerase at the start site of transcription on the promoter, but it's a closed promoter. We have to open it up first, and then we can start transcribing. But the first thing that happens is not a smooth transcription. The first thing that happens is several abortive initiations. The polymerase is trying to break free of that promoter, but the sigma factor is still stuck to it, and that prevents the polymerase from escaping from the promoter. We have to get rid of that sigma factor first. So the sigma factor goes away, and if the core polymerase itself is now free to begin transcription. And so we have elongation as the polymerase goes down that DNA template. It's going to read this template 3' to 5', and it's going to synthesize the complementary RNA 5' to 3'. And so it's going along and it's elongating, it's synthesizing that RNA, and finally we have termination. How does that happen? There are two main types of termination for E. coli transcripts. There's one that requires Rho, and there's one that doesn't. The one that doesn't require Rho has a termination sequence that ends in a series of U's. Let's look at that first. What happens is a hairpin is formed. The RNA itself forms a higher order structure, secondary structure, by folding in on itself. There's a palindrome. We have G's on this side, or we'd have C's on the other, or vice versa, and they bind together through hydrogen bonding, and they form a hairpin structure. This is a stem loop structure. We have the hairpin that has C's and G's along the stem, and then a loop. And then in the 3' region, we have a series of U's. Now, these U's are bound, of course, by hydrogen bonds to A's in the template, but these UA base pairs are weak. So we have strong hydrogen bonding in the hairpin. We have weak hydrogen bonding between the U's and the A's in the template, and we have a paused polymerase that has formed this hairpin, and it can't progress, and it's kind of stuck there. And so we have some tension that's building up. And so how is this tension relieved? Are the C's and G's going to break first, or the U's and A's? The U's and A's are going to break. The RNA with its hairpin is going to fall off the template. The polymerase is going to fall off the template too, and transcription is going to stop. So that is row-independent termination, or it's also called intrinsic termination because that's intrinsic to the actual transcript itself. It's written right there in the message. Nothing else is needed to terminate this transcript. But there are some RNA transcripts that require something else. That's because their termination sequence doesn't have a series of just U's. There are other ribonucleotides there as well. And so that 3' end of the RNA is going to be stuck to the template a little bit stronger than what we saw before. So we still have a hairpin that's formed, but we have strong base pairing here. And so when that tension builds up, something has to give, right? We have to find some way to pop that transcript off of the template. This is where Rho comes along. Rho is a multimeric protein that looks sort of like a donut, and it is an ATPase. So it can bind to a region of the transcript that's just come out of the polymerase and can attach there, and then through ATP hydrolysis, it can move up that transcript until it hits the paused polymerase to dislodge the RNA from the template. So the RNA falls off, polymerase falls off, and we have termination. And again, that's row-dependent termination. So we said that bacteria like E. coli do not do a lot of post-transcriptional modification, and this is true, but they do a little bit. For instance, when they transcribe tRNAs, several are synthesized as one long RNA that's then clipped. Those tRNAs have to adopt a very complex higher order structure. And so they need a little help to do that, to have a little bit more stability. Some of the ribonucleotide bases are going to be modified. So for instance, here we see uridylate. That can be modified to something called pseudouridylate or ribothymidylate. Do you need to know those names? No, I promise you don't. What you do need to know is that following the transcription of tRNAs, some of the bases are changed in order to provide more structural stability to the tRNA. Why? Because these particular bases can form different and stronger hydrogen bonding to form stronger base pairing to help keep that tRNA in the proper shape. Another thing that happens is we're going to remove the 5' leader sequence and also part of the 3' end as well. And we're going to add a CCA. This is going to be used as an attachment site for amino acids. And so we're going to talk about synthesis of rRNA. This is ribosomal RNA that's going to be used to make ribosomes. Ribosomal RNAs are synthesized as one long strand of RNA that's then clipped into shorter segments. And then these liberated RNAs can go and make ribosomal RNA and tRNA. And so that clipping is something that E. coli do. And base transformation is something that E. coli do. But they don't do the splicing that we do, and they don't put a cap and a tail on their messenger RNA like we do either. We're going to look at that in just a minute when we look at eukaryotic transcription. So E. coli do some things, but they don't do much. So let's move on to eukaryotic transcription. That is much more complex, and we're going to talk about the regulation of this transcription later on. We're going to talk about a lot more transcription factors in the next lecture. But for now, we're going to talk about something that's called basal transcription. And this is the non-activated type of transcription that happens. Not an activated transcription, though. In activated transcription, that means a specific thing. That means that you have something else available besides the basal transcription machinery that's going to additionally help the polymerase to transcribe this gene more often. And so that's activated. Basal just means it's the low normal rate of transcription for that gene. An activated transcriptional rate means it's a higher rate than basal. So to get basal transcription, you have to make what's called a pre-initiation complex. And so there is a small cohort of transcription factors that are necessary to get this basal non-activated transcription going. So we have 3 main polymerases, Pol1, Pol2, and Pol3. These have different roles, and they're associated with different transcription factors, and they use different promoters. We're going to concentrate on Pol2 transcription, but let's talk about what Pol1 and 2 and 3 do. The Pol-1 RNA polymerase transcribes ribosomal RNA. This is 18S, 28S, and 5.8S. RNA polymerase 2 transcribes messenger RNA and also SNRPs. We'll talk about those in a minute when we talk about splicing. Those are small ribonucleic acid protein particles. So, Pol2 transcribes messenger RNA and SNRP RNA. Pol3 transcribes tRNAs and also 5S ribosomal RNA. How did they figure out that there were several types of RNA polymerases? Well, first they were able to resolve these different polymerases on an electrophoresis gel. But second, they decided they were going to poison them by using alpha-amanitin. This is a toxin that's made by the toxic mushrooms, Amanita phalloides. This is a mushroom that's responsible for a lot of poisonings every year because it looks a little bit similar to some other mushrooms that are edible, but these ones are not. These ones will kill you, and that's why they're called the death cap mushroom. And these 3 polymerases have different sensitivities to this poison. For instance, Pol-1, not very sensitive at all. Pol-2, strongly inhibited. And Pol-3, that's inhibited, but you have to throw a lot of alpha-amanitin at it to inhibit it. Do you have to know the susceptibilities of each of these polymerases to alpha-amanitin? No, you don't. I'm just sort of giving you some background to tell you how they found out about these things, because biochemists are a strange lot. They generally find out a lot of their information by throwing a wrench in the system and seeing what breaks. Don't let anyone fool you. That's how biochemistry is done. So eukaryotes have different polymerases. They also have different types of promoters that are recognized by these polymerases. Do you need to memorize all of these different types of promoters? No, you sure don't. I just want to show you that there are different types of promoters for each of these polymerases. Some have recognition sequences that are way 5' to the start site of transcription. Some have promoter sequences that are actually downstream of the start site of transcription. And so there's a lot of variability in eukaryotic promoters. We're going to talk about Pol2 promoters extensively. In fact, we're going to talk about specific types of Pol2 promoters, those that require TBP. Those are promoters that include what's called a TATA box, TATA, and that's because of the sequence in that region. So let's look at initiation in eukaryotes and the making of the pre-initiation complex. As you would probably expect by now, the pre-initiation complex in eukaryotes is much more complex than what we saw with bacteria. The Pol2 pre-initiation complex consists of transcription factors that are shown here in blue, and also the polymerase that's brought to the start site of transcription. So we're going to have several basal transcription factors that are going to help target the polymerase. Some of those transcription factors are going to find the promoter first and then recruit the polymerase to the start site. Let's see how that happens. So the first thing that's going to happen is a protein called TBP, TATA binding protein, is going to bind to the TATA box. This TATA binding protein is a slightly asymmetric protein that looks like a saddle. It looks like it has a seat and the stirrups coming down. The stirrups actually include phenylalanine that intercalate between base pairs to bind really effectively to the DNA and also to change its architecture. Now, when you think of a saddle, what do you think of? You think of a saddle as binding to the back of a horse, like this, right? You can imagine that the saddle would sit down on the fiber like a saddle on a horse's back. No, that's not what happens at all, though, it turns out. This is pretty wild. I have a wild story for you. When I was a graduate student at Yale, these postdocs in the Sigler lab just down the hallway from me were actually figuring this out. In fact, Dr. Geiger in the chemistry department was one of the people who helped to do this. They had the protein structure pretty well in hand, but then they started modeling the DNA in, and there was an awful lot of excitement all of a sudden. People were running down the hallway to actually see what was going on, and it was wild. These people were just absolutely gobsmacked. Why? Because the TBP wasn't sitting on the horse's back like this. No, it was bending the horse. It was actually deforming the DNA, bending it. Phenylalanines in these stirrups go between the base pairs and sort of apply a little bit of pressure and bend that DNA. And this does a lot of things. This helps for initiation for the unwinding of the DNA. This makes it easier for other transcription factors to bind to the DNA and to melt those strands. Those transcription factors that do that are going to come later on. But TBP actually sets the stage. It's a remarkable protein. TBP is actually part of a larger complex that's called TF2D. So when TBP binds, it binds with its friends. Its friends are TBP associative factors. These are called TAFs. And so TBP and its TAFs are called TF2D together. So TBP and its friends make TF2D. The next protein to bind is going to be TF2A. It's going to bind just upstream to TBP. It can find the right place to bind because TBP is a little bit asymmetric, and so it has some directionality. But this directionality is further reinforced by the other transcription factors that are going to bind. And then there's TF2B downstream. And this helps to orient the polymerase when it comes in so that it goes in the right direction and it's actually going to use the right strand as a template. So we have TBP and TF2A and TF2B. Why are they called TF2A and TF2B? Good question. TF means transcription factor. 2 means we're going to be using polymerase 2. And A or B or D is the actual name of the transcription factor. So TBP and its friends make TF2D. They recruit TF2A upstream, TF2B downstream, and then TF2E and TF2F, and finally TF2H. Those are the main ones. TF2E and F are responsible for helping to recruit that polymerase and have it bind effectively. TF2H is a really big, really important basal transcription factor. This has a helicase capability, so it helps to unwind that DNA, and it also has a kinase. This turns out to be really important because the polymerase has a C-terminal domain. Remember how alphas had C-terminal domains? Well, this polymerase has a C-terminal domain also. So what's going to happen is, first of all, we have the pre-initiation complex at the start site of transcription on the promoter, but it's a closed promoter. We have to open it up first. TF2H has a helicase capability, so it helps to unwind that DNA, and TF2H is going to phosphorylate that C-terminal domain, and that polymerase can leave the promoter. So now we have promoter escape, and then we can go on to elongation. It's pretty cool. This C-terminal domain helps to recruit enzymes for capping the mRNA, for splicing the mRNA, and for terminating and putting a 3' polyadenylate tail on that message. Let's talk about the things that happen during transcription in eukaryotes. We're going to talk about 5' capping, splicing, and termination plus polyadenylate tail formation. So let's do that. The mRNAs in eukaryotes contain a 5' cap and a 3' polyadenylate tail, and these serve several purposes. One of them is to protect the message itself, to sort of cap both ends so that it doesn't become degraded by exonucleases. The other thing that these two regions do, the cap and the tail, is they can bind to other proteins to circularize that message. The 5' cap is also a place for the ribosome to bind to. So during translation, the ribosome is going to bind to the 5' cap and find the right place to start translation. So 5' cap, 3' polyadenylate tail are used for protecting the message, circularizing the message, and also the 5' cap is used for ribosome binding. So as the mRNA itself is synthesized, the cap is going to be added. You need to go into the whole process of capping? No, you don't. What you do need to know is that this is a guanosine cap that is methylated. We're going to add a guanylate to the 5' end of this message, and then we're going to methylate that. This is the 5' methylguanosine cap. And that's all you need to know about that. So how do you form the tail? Well, there's a cleavage signal that's produced in the messenger RNA itself. This is going to be recognized and cleaved by endonucleases. And then ATP is going to be used as a source of adenylate. And these adenylates are going to be added through the action of a poly-A polymerase to form the poly-A tail. And in this figure, those are shown as blue. The other thing we have to talk about is splicing. This splicing is carried out by U-SNRPs. U1, U2, U4, U5, U6. You need to know these numbers and what they do? No, you don't. You just need to know that these are U-SNRPs that are going to do this, and they're comprised of RNA and protein. The RNA is catalytic and the protein is structural. So what do you need for splicing? You need the U-SNRPs, and you also need 3 specific regions in the RNA itself. These 3 regions are a 5' splice site, an adenylate branch site, and then a 3' splice site. So a 5' splice site, a 3' splice site, and then in the middle, an adenylate residue that's going to be really important for starting the whole thing going. So what happens? The first thing that's going to happen is that adenylate branch site is going to make a nucleophilic attack on the 5' splice site. But it's a very strange thing that happens actually. The 2' hydroxyl is going to make that nucleophilic attack and form a phosphoester bond to that guanylate. So we're going from one phosphodiester bond to another one. But this one that's made is being made to the 2' hydroxyl on that branch site. So what this does is it actually frees up that exon number 1 that's shown in blue. Now it has a 3' hydroxyl that can do a little damage. It goes and it makes a nucleophilic attack on the 3' splice site. And what that means is now we have exon 1 and exon 2 spliced together now. And that intermediate, that lariat loop region is out of there. That's gone. When this 2' hydroxyl makes a nucleophilic attack and makes this phosphoester bond to the guanylate, it actually forms sort of like a loop, a lariat structure. And then the exon number 1 is free to make a nucleophilic attack on the 3' splice site. So now these 2 regions are together, exon 1 and 2 are together, and there's a bond that's been formed, and the lariat itself has been released. And that's how it's done. So what about Pol-1 and Pol-3 transcription? In Pol-1 transcription, we're going to make what kind of RNA? Going to make ribosomal RNA. How do I remember this? I have a really good way of remembering this. You know how in translation you have the ribosome that comes first, and then you have the messenger RNA that comes to the ribosome. So ribosome first, then messenger RNA, and finally tRNA. Ribosome, Pol-1, messenger RNA, Pol-2, tRNA, Pol-3. 1, 2, 3. That's how I remember it. Pretty cool. So anyway, Pol-1 transcription, we're going to synthesize ribosomal RNA, and again, that's synthesized in one long chain for 18S, 28S, 5.8S. And then these rRNAs are clipped out, and the bases again are changed in order to make them more structurally sound. With Pol-3 transcripts, with tRNA specifically, we're going to do a lot more than that. We're going to first take the primary transcript, the early transcript, and we're going to splice out part of the bottom loop to form the anticodon. And then we're going to remove the 5' leader sequence and also part of the 3' end as well. So we're going to clip off the 5' region and the 3' region. And to the 3' region, we're going to add a CCA. This is going to be used as an attachment site for amino acids. And so we've spliced, we've clipped off the 5' leader, we've clipped off a portion of the 3' region, and we've added a CCA region to make the amino acid attachment site. Another thing we do is, just like in bacteria, we're going to change some of those base pairs in the tRNA, again to provide structural integrity to the secondary structure of the tRNA so that it can make those stronger hydrogen bonds and hold the structure together. So this marks the end of lecture 37. I hope you enjoyed it. I hope you learned a lot. and I hope you have a great day.